Hi everyone, and welcome to today's Daily Booster, which is number 8 in our series. First thing we're going to look at today is our daily formula, of course, and today's daily formula is for power. So power in watts can be calculated by doing the work done in joules divided by the time in seconds. For today's Biology Blast, we're going to be having a look at the development of new medicines in terms of drug trials. So this comes up in B6 and therefore is going to be the second biology paper. When we've actually identified something we think could be a new drug, then we need to test it in a lab to see how it behaves before we release it on the general public. So the first thing we do is something called a preclinical trial. Now, preclinical trials are where we're actually going to be having a look to see how that drug impacts on things like a dish with live cells, or potentially bacteria or tissue culture. So the idea of these preclinical trials is to make sure that when it is exposed to cells, it doesn't just kill them all. Because obviously we don't then want to introduce that drug to an entire living being, because it would end badly. If we pass the preclinical trial, then we move on to the next stage in testing, which is the animal testing. Now, for animal testing to actually be passed, it must actually pass with 2 different species before we can look at human trials. The idea behind that being that if you only test it with a single animal species, then there is the risk that it may not behave in a similar way in all animal species. So testing with 2 just gives us a slightly greater range there. Because animal testing is not always the best thing, we do have our 3 R's principle to try to reduce the impact of this. First R, reduction. We use the smallest number of animals possible. Second R, refinement, improving the experiments to avoid any unnecessary suffering and also to improve animal care for any of the animals in these test facilities. And the third R, replacement, replace the animals with other techniques where possible. If we pass our animal trial, we then move on to the clinical trials, which is where humans become involved for the first time. We've got 3 stages to these human trials. Step 1 is healthy volunteers. We use the healthy volunteers in order to look for any side effects with using that medication. Step 2 is we have a small sample of volunteers that have our condition, and the idea behind that is just to see how effective our new treatment actually is at treating said condition. And then step 3, we increase the number of volunteers with the condition, so we've got a large number now to test the effectiveness and also to check for the safety. So we're reporting any side effects, we're checking how effective it is in a large number of people to build up that evidence base. When we're carrying out these trials with humans, then we sometimes use a type of drug called a placebo. Now, a placebo is basically a sugar pill. It's a medication that has no active ingredient in it. So the idea behind this is we can carry out trials where people aren't certain if they're getting the actual medication that's being tested or if they're getting the placebo, which in theory does nothing. Reason we do this is because of this thing called the placebo effect. Now, the placebo effect is basically where if you give people a medication or something and tell them this is going to make you feel better, then people start to feel better because they expect to, not because of anything you've given them actually doing a thing. So the placebo effect is basically a psychological mindset whereby people think they're getting better just because they believe they should be. So what we use is something called a double-blind trial. Now, a double-blind trial is where we will have some patients getting the drug while others get the placebo. But the key thing about this and why it's called a double-blind trial is because neither the patients nor the doctor who's administering the test know who's getting which one. So that removes any element of bias from the whole procedure. Quick reminder before we move on to chemistry of our daily formula. Power in watts is the work done in joules divided by the time in seconds. For today's chemistry check, we're dropping back to C4, which is on paper 2 for chemistry, and we're going to look at the noble gases, which is group 0 on the periodic table. As before, do remember in every chemistry exam you have a periodic table, so you don't have to memorize the names or the symbols. Just remember group 0, far right hand side, those are the noble gases. The reason we call them the noble gases is because they are unreactive. So these are the ones that really don't take part in many chemical reactions at all. A few other key facts about them. They are non-metals because they're on the right hand side of the periodic table where all the non-metals are. They are all gases at room temperature. The name the noble gases should tell us that bit. And the reason that they're all unreactive is because they all have full outer shells of electrons. And as we've seen previously, the reactivity is linked into how easy it is to gain or lose electrons. In this case, we've got a full outer shell. They're really stable. That means they're unreactive. So if ever you get a question asking why the noble gases are unreactive, because they have a full outer shell of electrons is our answer. Because they've got that full outer shell of electrons, then they go around as what's referred to as monatomic. So they are single atoms with very weak forces of attraction between them. Hence, they're gases. There are a couple of patterns we need to remember as well. As we go down group 0, then we get larger atoms. The larger the atoms, the stronger the attractive force will be. Because the attractive force gets stronger between those atoms, then their boiling point will increase, and they're also going to have an increase in density the further down the group we go. So just remember those three patterns. Going down, then obviously the atoms are getting larger, so the attractive forces between the atoms get stronger, the boiling point will increase as a result of that, and also density will increase. Quick reminder of our daily formula again. Power in watts is the work done in joules divided by the time in seconds. For today's physics prep, we're going to look at transformers. Now, transformers are something that come up in P4, and they will also come up again later on. We are not talking about Transformers from the cartoon or the movie franchise. We are not talking robots in disguise. We are talking about structures that increase or decrease the potential difference. So, how do transformers work? Hopefully we remember that potential difference is induced when magnetic field lines cutting a coil change. If we can change the field lines that cut a coil, then we're going to induce a potential difference. And there's three ways that we can change those field lines. The first one, we can move the magnet or the coil. The second is we can use another coil to turn the current in it on or off. Or the third one, we can use another coil with an alternating current. If you remember, alternating current just means it constantly switches between the positive and the negative. Therefore, it's a constantly changing current. The actual structure of our transformer is at the bottom there. So what we have is this iron core, so a loop of iron basically, and we've got two coils. On the left, we have our primary coil, and on the right, we have our secondary coil. So that as we're having a current flow through the primary coil, we're then going to generate a current in the secondary coil, all based on this idea of inducing potential difference. So how does it work? What we have then, an alternating potential difference across the primary coil, the red one on the left-hand side there, is going to produce an alternating current in that primary coil itself. So the alternating potential difference produces an alternating current in the primary coil. Because we've got an alternating current in the primary coil, we have a magnetic field being produced in the iron core, which is always changing. Because remember, if it's an alternating current, it's constantly flipping between the positive and the negative, therefore that's going to cause a changing magnetic field. The iron itself is obviously a loop, so that means the whole of the iron is going to have that magnetic field, and therefore we're going to induce a changing potential difference in the secondary coil. Because if we've got a magnetic field that's constantly changing, then obviously that coil of wire is going to have an induced potential difference in it, in the secondary coil. So in terms of why we use these transformers then, we will see them come up again when we get onto P8 on the second physics topic in the national grid. We generally have two types of transformers we use, step up and step down. A step up transformer increases the potential difference. And the way it does that is that we've got more turns on the secondary coil than we do on the primary. So you can remember it that we're stepping up the number of coils, which means we're stepping up the potential difference. The step down transformer is going to decrease the potential difference. And it does this by having more turns on the primary coil than on the secondary. So we're reducing the number of coils as we go from primary to secondary, which means we're also decreasing the potential difference. Hopefully you found that useful, and don't forget we'll be back again tomorrow for the last one of our daily boosters for this season.